Gene or allelic interaction is simply the communication between the alleles of the gene or genes in such a way to result a distinct phenotype depending upon the nature of the gene or genes whose alleles are involved in the communication. The alleles involved in the interaction may either belong to same gene called intragenic interaction or two different genes called intergenic allele interaction. The intragenic interaction include the normal Mendelian phenomenon of dominance and recessiveness, incomplete dominance and codominance, while intergenic interaction involves the complementary gene interaction, supplementary gene interaction, epistasis, pleiotropy, multiple alleles, duplicate genes, and lethal gene interaction. It was Gregor John Mendel 1822 to 1884 who performed experiments with garden pea and came to the conclusion that every character is controlled by a pair of factors and led the principles of heredity, which in turn has laid the foundation of the genetics. Mendel identified two alleles, one dominant and other recessive. This discovery suggests a simple dichotomy between alleles, as if one allele did nothing and the other did everything to determine the phenotype. However, the research early in the 20th century demonstrated it to be an oversimplification. Genes can exist in more than two allelic states, and each allele can have different effect on the phenotype. Depending upon the nature of the alleles involved in the interaction, gene interaction is classified into two types, viz. Intergenic interaction and intergenic gene interaction. A. Intragenic gene interaction. As the name indicates, the alleles involved in the intragenic interaction belong to the same and result a distinct phenotype. The examples of such interaction include dominance recessive interaction, incomplete dominance, and codominance. Dominance recessive interaction. It states that when two contrasting alleles of the same gene come together in an individual, only one of the alleles expresses and other remains dormant. The allele which expresses is called dominant allele, whereas the allele that remains dormant is called recessive allele. For example, when Mendel crossed a homozygous pea plant with red flowers with a plant having white flowers, all the F1 individuals were with red flower, and in F2 generation the ratio of red to white was 3:1. This indicates that the allele for pink color is dominant, whereas allele for white color is recessive. Number second, incomplete dominance. Incomplete dominance is the phenomenon where none of the two Mendelian factors or alleles is dominant over the other. The expression of the character in the hybrid is intermediate between the expression of the two factors in their homozygous state. For example, in Mirabilis jalapa, there are two types of flower colors, red and white. When the two types of plants are crossed in F1 generation, they produce all pink plants, showing that neither red nor white genes are dominant. The pink color appears to be formed by mixing of red and white colors. When the hybrids of F1 generation are crossed, they produce the plants which are of three types in the F2 generation, that is, red flowered, pink flowered, and white flowered in the ratio of 1:2:1. This genotype shows that pink color is not produced by any specific gene but the joint activity of two alleles. Same is the case in Andalusian fowls, where homozygous black and homozygous white produces blue colored hybrids in F1 generation. Here blue color appears due to fine mixing of black and white stripes on the feathers. Number third, codominance. Codominance is the equal and independent expression of the two alleles of a trait when they are present together in an individual. Instead, both the alleles are able to express themselves when they occur together. The phenotype of hybrid individual is usually different from either of the homozygous phenotypes. There is no mixture of the two traits, though they may occur side by side. Because of this fact, the appearance of roan color in hornless cattle is often considered to be a case of codominance. The symbols for codominant genes are different. Here, uppercase or capital base symbols are employed for both the alleles but with different superscripts, example IA, IB. In human beings, genes for blood groups show both codominant and dominant recessive relationships. As the name indicates, the alleles involved in the intergenic interaction belong to the two different genes and result a distinct phenotype. The examples of such interaction include duplicate genes, supplementary genes, complementary genes, lethal genes, epistasis, and multiple alleles. Number 1. Duplicate genes. Duplicate genes are two or more independent genes present on different chromosomes which determine the same or nearly same phenotype so that either of them can produce the same character. The independent genes do not have cumulative effect. They produce the same phenotype whether present in homozygous or heterozygous state. For example, endosperm color in maize is of two types, yellow and white. Yellow color is dominant over the white color. Yellow color is determined by one or two independent dominant genes, Y1, Y2. Their recessive alleles give rise to white endosperm. When a plant with pure yellow endosperm is crossed with white colored endosperm, the F1 hybrid will produce grains with yellow endosperm. On selfing in F2 generation, they produce grains with both yellow and white endosperm in the ratio 15:1. It is suggested that the number of dominant genes do not make any difference as there is no difference in the yellow pigment of grains of various plants. 1 out of the 16 progeny homozygous dominant for both the genes, 6 homozygous dominant for one of the two genes, 4 heterozygous dominant for both the genes, 4 heterozygous dominant for one gene, while 1 is homozygous recessive for both the genes. Number 2. Complementary genes. Complementary genes are those non-allelic genes which independently show a similar effect but produce a new trait when present together in the dominant form. Flower color in sweet pea. Sweet pea has two strains of white flowered plants. If the two pure breeding strains are crossed, they produce plants with purple colored flowers. When F1 hybrids are crossed, they produce both purple and white flowered plants in F2 generation in the ratio of 9:7. The appearance of purple color in 9/16 population shows that the color is determined by dominant alleles of two genes. When either of the two is absent, the pigment does not appear. It is believed that the dominant gene C produces an enzyme which converts the raw materials or substrate into chromogen. The other dominant gene P gives rise to an oxidase enzyme that changes chromogen in a purple anthocyanin pigment. This is confirmed by mixing the extract of the two types of flowers when purple color is formed. Thus, purple color formation is a two-step reaction and the two cooperate to form the ultimate product. Number 3. Supplementary genes. They are a pair of non-allelic genes, one of which produces its effects independently when in dominant state, while the dominant allele of the other is without any independent effect but is able to produce a new trait along with the dominant allele of the former. Coat color in mice and guinea pigs. The animals have three types of coat color: agouti, black, and albino. Albino animals have white coat color which is produced by a recessive gene, whereas the dominant genes produces black coat color animals. There is another non-allelic gene which when dominant changes the expression of black coat, forming dominant gene into agouti or grey coat color. Agouti color is produced by the presence of black pigment in the hair with a strip of yellow color below the tip. When a pure black mouse is crossed with a pure albino mouse, they produce agouti hybrids in the F1 generation. But when F2 hybrids are cross-pollinated, they produce agouti, black and albino animals in the ratio of 9:3:4. It is the phenomenon of masking or suppressing the expression of a gene by another non-allelic gene. The gene which suppresses the expression of a non-allelic gene is known as epistatic genes. The gene or locus which is suppressed by the presence of non-allelic gene is termed as hypostatic gene. However, it has been found that epistasis is not a simple phenomenon of one gene suppressing another. Both the non-allelic genes may be mutually epistatic to each other. Epistasis is of three types. Number 1, dominant epistasis. Number 2, recessive epistasis. And the third one is dominant recessive epistasis. Now, dominant epistasis. In dominant epistasis, the dominant gene at one locus suppresses the expression of another gene at a different locus regardless of its allelic conditions. Fruit color in cucurbita pepo. In cucurbita pepo, there are three types of fruit colors: yellow, green and white. White color is dominant over other colors, while yellow is dominant over green color of the fruit. Yellow color is formed only when the dominant epistatic gene is represented by its recessive allele. When the hypostatic gene is also recessive, the color of fruit is green. When a pure white cucurbita pepo plant is crossed with a pure green cucurbita pepo plant, they produce hybrid white fruit color plants in F1 generation. On selfing, F1 plants produces three types of fruit colors in the F2 generation as 12 white, 3 yellow and 1 green fruit color plants. Number 2 is recessive epistasis. In recessive epistasis, a recessive homozygous genotype at one locus suppresses the expression of non-alleles at another locus. The latter can produce their effect only when a dominant allele occurs at the first locus. Pigmentation in onion bulb. Onion bulb can have three types of pigmentation: red, yellow and white. When a homozygous red onion is crossed with a homozygous white onion, it produces red-colored onions in F1 generation. On self-pollination, F1 hybrids produces three types of onions in the F2 generation as 9 red, 3 yellow and 4 white. The appearance of white color is due to pigment inhibitor gene, which is epistatic only in the recessive state. In the dominant state, the gene allows the pigment-producing gene to express its effect. Yellow color is formed by the residual genotype or recessive alleles of the pigment-producing gene. Dominant recessive epistasis. In the dominant recessive epistasis, the dominant allele at one locus and recessive allele at another locus give rise to the same effect. Other gene combinations produce the second phenotype, so consequently two phenotypes occur. Plumage color in poultry. Here, white plumage of Wyandotte or Plymouth cock is recessive to colored plumage, while white plumage leghorn is dominant over colored plumage. When pure white leghorn is crossed to pure white Wyandotte, it produces white plumage in F1 generation. In F2 generation, white and colored plumage are produced in the ratio of 13:3 instead of 12:3:1 because the recessive epistatic gene also produces the same phenotype as dominant epistasis. Lethal genes are those genes which kill the organisms when they are able to express their effect. They can be dominant or recessive. Dominant lethal genes cannot exist in the population as they would cause the death of the possessor even in the heterozygous condition. Thus, the existing lethal genes are either recessive or become lethal only in homozygous state. They occur in the population in the heterozygous state because their effect is kept masked by the normal alleles. Even in heterozygous condition, Some of the lethal genes reduce vitality and affect viability. Example, sickle cell anemia under stress, curly wings in Drosophila. Lethality. Lethality is the effect of lethal genes in causing the death of the individual. Depending upon the period when lethal genes become fatal, lethality is of three types. Absolute lethality, here the individual dies in the embryonic stage. For example, yellow fur in mice. Sublethality, when the organism dies before the attainment of reproductive age. Example, sickle cell anemia. Delayed lethality, when the organism dies after the attainment of reproductive age. Lethal genes disturb the monohybrid ratio of 3:1 or 1:2:1 in the F2 generation because 1/4 progeny becomes homozygous for the lethal gene that kills the bearer. The surviving individuals have a ratio of 2:1 or all of them are phenotypically similar. Sickle cell anemia. Sickle cell anemia is an autosomal hereditary disorder in which the erythrocytes become sickle-shaped under oxygen deficiency. The disorder is caused by the formation of abnormal hemoglobin S. Erythrocytes having hemoglobin S become sickle-shaped under oxygen stress as during strenuous exercise and at high altitude. The cells also have a tendency to clot and degenerate. They clog blood capillaries, disturb blood circulation, reduce supply of oxygen, food and other substances. Spleen and brain get damaged. The homozygous individuals with only hemoglobin S usually die before reaching maturity. However, heterozygous individuals with both hemoglobin A and hemoglobin S appear normal till there is oxygen deficiency. It changes some erythrocytes into sickle shape. The gene for sickle-celled erythrocyte is HbS, whereas that of normal erythrocytes is HbA. The homozygotes for the two types are HbS and HbA respectively, whereas the heterozygous form is HbA-HbS. When two sickle cell carriers or heterozygotes marry, they produce three types of children: homozygous normal, heterozygous carrier and homozygous sickle-celled in the ratio of 1:2:1. However, homozygous sickle-celled individuals die in the childhood due to acute anemia. Thus, a ratio of 1 normal to 2 carriers is obtained. Number 6 is suppressor genes, which is also known as inhibitor genes. Inhibitor gene is a non-allelic gene which does not produce any phenotypic expression of its own but suppresses the expression of a non-allelic gene. Leaf color in rice. In rice, the leaves can be purple or green. The green color can be due to presence of a recessive allele P and purple color due to presence of a dominant gene P. However, suppressor or inhibitor gene I suppresses the purple phenotype and causes formation of green leaves, whereas its recessive gene I does not have any suppressor effect. When two homozygous plants having green leaves are crossed, they produce hybrid green in F1 generation. But when F1 hybrids are self-pollinated, they produce green and purple-leaved plants in the ratio of 13:3 in the F2 generation. A gene has usually two alternate forms that represent the alternate expressions of a trait. They occur at the same gene locus on the homologous chromosomes. Certain genes have more than two alternate forms or alleles, which are termed as multiple alleles. Thus, more than two alternate forms of a gene that occur at the same gene locus and express the different phenotypes of the trait are termed as multiple alleles. The different alleles of a gene actually represent its mutant forms. For example, a gene A mutates to form allele A, where both A and A can mutate further to produce A1, A2, A3, A4, etc. The symbols employed for multiple alleles are slightly different. The wild, normal or dominant allele is designated by capital letter or lowercase letter with the sign of positive as superscript A or A+. The allele recessive to all others in multiple allele series is assigned the small letter without any superscript A. Other alleles are intermediate between the two and are represented by the lowercase letter with suitable superscript, which is AA, AB, AC, ACH, etc. In case two or more alleles are equally dominant over other alleles, capital letter with suitable superscript is assigned to them. For example, IA, IB, S1, S2, S3, etc. In self-sterility, presence of a particular sterility allele in ovule mother presents the germination and growth of pollen grain having the same allele. The gene for self-sterility of red clover has about 200 alleles. Human blood groups. On the basis of presence or absence of certain antigens, ABO blood groups have been established in man. It was found that there can be two antigens A or B, and as a result 4 blood groups called ABO blood group or Landsteiner blood groups. If donor's blood has antibodies against that recipient, it does not matter since they get diluted. However, there should be no antibodies in recipient's blood against donor's blood, since this will cause agglutinization of transfused blood. As AB is universal recipient, so no test of donor's blood is necessary. Whereas O group is universal donor, so no test for the recipient's blood is necessary.